in parallel lives. And there, actually, he continued working on store-operated calcium entry, driving his own research program, and he became more and more focused on the role of STIM and ORI protein in certain types of cancers and the regulation of STIM by EGR proteins, and also some work on immune cells and the role of PMCA in shaping the calcium signal in those cells. And I'm not sure what he will be talking about today, but he recently published a couple of papers on this last part, and I know he's been also interested in working on these proteins called POST. And so John is a full professor, tenured, is very well funded, and without due delay, I'm just going to stop here and let John give his presentation.
All right. Well, thanks a lot. Thank you very much, Mo. Emily and Martin for the invite, and Mo for the nice introduction. I wonder if all that stuff could even be true that you said. the only one I probably have to correct is University of Ottawa, where I actually got my PhD. But in any case, yes, thank you very much. So actually, I'm going to talk today about melanoma progression. So this has been a slow-burning project in my lab for a number of years. Mo's right. We did publish a couple of papers in the last 12, 14 months on a couple of other topics. But I always like to talk about the new thing that we haven't published. It's kind of a dangerous habit, but I haven't yet been able to shake it. So let's get going. So I'll start off talking a little bit about store-operated calcium entry. I know some people are very familiar with it, and other people, work in other aspects of calcium signals. I'm gonna get the mouse. This is a cell and the molecular mechanisms that regulate store-operated calcium signals. So the colors are set to show the relative concentrations of calcium in the cell. So calcium is very high outside the cell and within the ER lumen, but much lower within the cytosol. So that's sort of described through this scale. Now on the left we have a cell at rest, and on the right we have a cell in which the store-operated calcium signaling pathway has been activated. So you can see on the left you have lots of calcium in the ER, and you have these STIM proteins. There's two, STIM1 and STIM2, which exist as resting dimers on the ER membrane, and they have these a luminal portion and a cytosolic portion. So the luminal portion of the protein has a calcium sensor, an EF hand that's in this resting state would be bound to calcium in the high luminal calcium concentration, and the protein is essentially at rest. Then we have these ORI channels in the plasma membrane, also at rest, no closed essentially. Now, any signal that depletes the ER of calcium can activate the signaling pathway. Physiologically, that typically would happen in response to a PLC-coupled receptor, such as shown here. And so about half of the GPCRs and tyrosine kinase-coupled receptors are all PLC-coupled receptors. And upon activation, PLC will cause the release of IP3 to stimulate calcium release from the IP3 receptor. And so when calcium leaves the ER and enters the cytosol, there's a decrease in ER calcium concentration. And that's sensed by these STIM proteins, which are then activated. They lose their calcium. They form this extended conformation, and they form these large clusters that will then physically couple with ORI channels in the plasma membrane and gate the channel. The channel then becomes on. And they'll tend to stay on until the ER calcium concentration is restored. So as Mo mentioned, in the early days when I was a postdoc in Don's lab, and then during the first few years when I was at Temple as a faculty member, we were mainly focused on studying molecular mechanisms of store-operated calcium entry. We tried to look at the differences between STIM1 and STIM2, and some of the alternative targets that STIM1 has. Of course, STIM activates ORI, but this is biology. STIM does other things as well. It can be activated through other mechanisms. We've also got quite interested in T cells, and we've done a lot of work in that area, especially as Mo mentioned, as related to the EGR family of proteins. I have another project that's been ongoing for quite a few years now, studying remodeling of bone. And it looks like that stem ori system is quite important in the differentiation of both osteoclasts and osteoblasts, and that's an ongoing project in my lab. Today I want to talk about melanoma. So this has been kind of a slow burning project. It's going on for quite a long time. Rob Hooper drove this paper in molecular cell biology from 5 years ago. Mo actually contributed to that paper, as it happens. It's our introducer. The project was kind of a slow burning state until I recruited Scott Gross, a graduate student in my lab, who took it over and has really driven it quite energetically. And pretty much all the data I'm going to show you today was done by him. So just a little bit about melanoma. I'm really just using this slide as an example of the etiology of melanoma. I don't really want to make a big point about these DNA mutations shown here. So what is melanoma? It's a cancer of melanocytes. And so melanocytes are a relatively rare cell type within the skin. I'm sure everyone realizes that they're the source of color. They're very prone to growing, to proliferate. They should not be proliferating and expanding, but they will do that. And so this can happen. This can be quite benign, but then of course benign growth can ultimately change into a mind blank, not benign state. Okay, and so a benign growth of the melanocytes can form what's called a nevus. I've had a number of them myself that can be quite easily removed. However, the nevus malignant is the word I was looking for. So when a nevus continues to grow more rapidly, becomes a little bit more dangerous and becomes a malignant melanoma. Initially, they grow in a radial growth phase, so that would be growth through the skin. The mole would simply get wider, and then it transitions into a vertical growth phase, which ultimately leads to metastasis and a much more dangerous disease. And so up until the vertical growth phase, this is a very easily treatable disease. A dermatologist just cuts the mole out. After the melanoma has metastasized, it's a much more dangerous disease, and there's still quite a few people that get this disease and die from it. So there is a need to understand the molecular mechanisms that drive this progression from the early to later stages. So we're interested in calcium signaling, generally speaking. A number of years ago, we started working on melanoma. Actually, the reason we started working on it was mainly because of some work that Rob Hooper was aware of, actually from Ashley Wehrer, who at the time was at the Wistar Institute. So actually had shown that there was a shift in Wnt signaling between non-invasive and invasive melanoma towards the non-canonical Wnt signal. And non-canonical Wnt signaling actually has been published extensively to work through calcium signals. And we thought, okay, well, why don't we look at these not invasive melanoma cells that express these non-canonical Wnt pathway activators. Wnt5A is the non-canonical activator I'm talking about. And then look at the calcium responses. To our surprise, we didn't really get what we expected, but that's okay because we then worked out to try to understand what it was that we were seeing. So when we looked at stropital calcium signals in these Wnt5A expressing invasive melanomas, we saw that they had really very small calcium signals, almost none. And whereas the non-invasive ones that didn't express Wnt5A had varying levels of calcium responses, but significant ones, much larger than the invasive ones. I'm not going to take you through the whole paper. It's published. It's almost 6 years old. But we were able to show that this calcium response was dependent on Wnt5A, and so if we overexpressed Wnt5A in the non-invasive melanoma, we would inhibit calcium responses, and if we knocked out Wnt5A in the invasive melanoma, we would stimulate calcium responses. We further showed that this Wnt5A expression was in fact driving invasion. And so we went on, and I'm not going to go through that paper, but essentially what we established was that stropital calcium entry was inhibited by Wnt5A, in that case actually through a phosphorylation-based mechanism. Happy to talk about it another time. All right, so we've been finished with that story. We did try to investigate the phosphorylation-based mechanism for a while, but in the end, we ended up changing tracks a little bit and thinking a little bit more about UV. We did this partly because of a collaborator very interested in UV, but also because UV, of course, is the primary driver of melanoma. I'm sure most people are aware of it. So there's three classes of UV, UVA, UVB, and UVC, but UVC doesn't actually enter the atmosphere, so it's not really relevant to melanoma. The UVB and UVA are. UVA can causes ROS damage, and UVB causes DNA damage. Interestingly, I mean, everybody knows this. Everybody knows that UV causes melanoma, but what interesting is that people don't exactly know why. And so, in an earlier slide that I gave you, I showed you. There's this list of these epigenetic modifications and spontaneous DNA mutations that are known to drive melanoma progression, but none of those genes are actually UV targets. And so exactly what it is that UV does that leads to those, leads to melanoma is far from clear. Further, although most people think about UV in the context of melanoma genesis, it turns out that UV also can drive melanoma metastasis. And so you can imagine a scenario where a person, where melanoma has formed, that person is still walking around in our world with the sun in it. And when that person goes outside, the UV, the radiation that that person receives, is further driving melanoma towards a metastatic state. And that's the feature that I'm gonna be talking about today. So what is the effect of how does UV affect drive metastasis in melanoma cells? Okay. So this is so the way we asked this question was we took a collection of cell lines that we knew were not invasive and did in fact have relatively normal looking calcium responses, and we looked at what effect UV radiation would have on them. Okay, and so when you radiate, so the way this experiment was actually done was the cells were UV irradiated with a dose that's approximately equivalent to what a melanocyte would see if it was in a person that was walking outside for about an hour in the sun. Okay, and so the cells were exposed to this UV radiation. They were then incubated for 24 hours, and we're not looking at immediate responses so much as what effect the UV has ultimately over time. This is a 24-hour time point, but we're actually going to move on from that. What we actually found is that essentially all the melanoma cells that we looked at and treated in this way exhibited significant decreases in store-beta calcium entry after a 24-hour incubation. Further, when we looked over time, it turns out that the store-beta calcium signals continue to change. So this is a somewhat stable or permanent effect. Okay, and so we see significant inhibition weeks after the fact. Although all of the cells showed an initial inhibition of calcium signals in response to UV, there were differences in how stable that response was, with WM983 cells showing relatively stable decreases in calcium signals, and the UACC257 cells showing a much later response than the WM983. It is strange the way the magnitude of the calcium signals seems to change over time. This stuff was repeated many times by Scott, and it seems to be what he sees. We actually tend to focus on this 2-week time point and the 5-week time points throughout the rest of this talk. So what I'm showing you here is that in fact, well, what I'm showing you is what one might expect to see if UV causes invasiveness, just like or melanoma progression, like I've shown you. So what we did is we treated cells. So cells were exposed to UV for 2 or 5 weeks, just like in this graph. And then we did a transwell and Bayesian assay. So this is a transwell assay through Matrigel. So what you see is that you do see a significant increase in the number of cells that invade after UV irradiation only under the conditions where we see inhibition of SOC, right? And so in the WM983 cells, where SOC was significantly suppressed at both 2 and 5 weeks. We see an increase in invasion. In the UAC 257 cells, where SOC was decreased at 5 weeks, but not 2 weeks, we only see increased invasiveness at 5 weeks. So there's a significant, there's a clear correlation between SOC suppression and increased invasiveness across these different cell lines in response to UV radiation. These were all human cell lines, but we wanted to look at mouse lines as well, not because we're particularly interested in the mouse, but rather because we wanted to do some in vivo studies, and we're going to do this in a syngeneic mouse model rather than trying to syngeneic. The immune system is actually quite a significant contributor to melanoma progression, and so we didn't want to use studies that excluded the immune system's contribution. So we do see somewhat similar inhibition of circulated calcium entry in B16 cells, although I do think this is a relatively weaker effect than what we're seeing in human cells. Nevertheless, it is significant, and we still see the increased invasiveness in this transwell migration, transwell invasion assay that we showed before. So since they respond somewhat similar, since B16 cells respond somewhat similarly to UV as human melanoma cells, we then went ahead and did the experiment that we were looking for. So the way this experiment was done was so B16 cells were injected subcutaneously into mice and then incubated for 2 weeks. So there are some B16 lines that are capable of metastasizing through subcutaneous mutation, but the B16N isn't one of them. So the control B16s don't metastasize, and you can see none of them actually left the primary tumor. They're all the cells just grow on the back of the mouse as a tumor. However, 5 weeks after, if the cells were UV radiated, incubated for 5 weeks, and then injected into the mouse, we saw 4 out of 5. I believe 4 out of 5 mice exhibited not metastasis, and so we saw tumors in the liver, lymph node, within the peritoneal wall, intestine, pancreas, stomach, kidney, and in one mouse, the spleen as well. You can see a picture of it here. So all this black material, that's all essentially the melanoma growing inside the mouse. Okay, and these are this is a tumor of the kidney, I believe. Oh, intestine and pancreas. Says right there, and this tumor is actually growing on the peritoneal wall. Okay, so what we see is that UV actually does drive melanoma metastasis in a syngeneic mouse model. Okay, so UV, well, I mean, we know that UVR does a lot of things. It causes ROS, it drives epigenetic changes, causes DNA damage. So next, what we wanted to know is, does SOC suppression itself have an independent effect on melanoma progression as a component of the UP response? So I haven't commented on it, but I'm sure many of you are aware that ORI is important, or the calcium signals are important in migration and metastasis. And there's plenty of studies showing that if you block stroke calcium entry, you will block metastasis. And we see essentially the same thing. This was actually the first experiment that we did. I believe this was actually started several years ago by Rob Cooper before Scott came. So if you block SOC, you block invasiveness in vitro and in vivo. I'm not showing the in vivo data, but it pretty much shows the same thing. So this is a dominant negative of ORI. It completely blocks stroke calcium entry, and cells expressing this ORI mutant failed to show any invasiveness in a transwell assay. However, as you've seen and as I sort of commented on, UV radiation doesn't ablate SOC. It suppresses SOC, and in some cases, somewhat modestly. Certainly in the B16 model, and it was a somewhat modest suppression. So maybe a modest suppression of stroke calcium entry does something quite different from what a complete ablation of stroke calcium entry would do. So the next thing we did is we did a titration of Btp2. Okay, so Btp2 is a pretty well-established inhibitor of stroke calcium entry. It's very effective. It works in a nanomolar range, and so you can see this is you can't really see it very well, but this is a This is a curve of, maybe we'll focus on this first. This is a curve of showing the effects of different concentrations of Btp2 on store-operated calcium signals. You know, I'm sorry, I never actually commented on it, but whenever I show these curves, these cells were treated with dapsogardin before you know, it was not shown. They were treated with dapsogardin in the absence of calcium. There was an ER calcium release, and then calcium was added back, and that's the part we're showing. Basically done this way so that the calcium add back point can are all lined up when we do overlays, right? Each one of these was done in a different cover slip, and the time after dapsogardin doesn't perfectly line up this way, unless shown in the way that I'm showing it. As a matter of reference, the effective the amount of store-operated calcium entry after a 5-week in cells that have been UV irradiated and incubated for 5 weeks is shown here. You can see it's much smaller than the control. And then you can see that actually the 40 and 80 nanomolar Btp2 concentrations show a relatively comparable inhibition of calcium signals comparable to the UV effect, with lower concentrations causing less effect and greater concentrations causing more. So interestingly, those same concentrations of Btp2, 40 and 80 nanomolar, caused a very significant increase in invasiveness in that same transwell invasion assay that I was showing you before. So partial SOC suppression does something really quite different from what total ablation of SOC actually does. That's really the point I want to make. Right. So we of course, we're interested in seeing whether or not this invasiveness can be translated into an in vivo system. This kind of titration of Btp2 concentration didn't seem realistic to me in an in vivo model, given you know, pharmacokinetics, the concentration of Btp2 wouldn't really be the same minute to minute, no matter what concentration that we used. We switched to a genetic model. Just briefly, we took advantage of, so it's been established that the overexpression of ORI wild type, not the dominant negative, will also inhibit SOC. That's essentially due to altered stoichiometry. I want to get into this picture again. This is basically stim at rest. Calcium depletion causes activation of stim, just sort of like what I showed you at the beginning, and in association with ORI. You notice that there's a lot of stim relative to ORI. It takes multiple stim dimers to fully activate ORI. The exact stoichiometry is known, but it's certainly clear that it's not one stim dimer per ORI, it's multiples. And so if there's lots of ORI, the probability of having enough stim per ORI goes down. So essentially what we did then is we created melanoma cell lines with ORI overexpressed, and then looked at how much SOC suppression we could get. And we went with these two, clones 3 and clone 44. So these are B16 melanoma cells overexpressing different amounts of ORI. We can't really control exactly how much ORI they actually have. The more ORI they had, the less store-operated calcium entry they got. And these two were relatively close to the amount of calcium suppression, store-operated calcium entry suppression that you would see in response to UV radiation, as shown here. Again, just like we saw with Btp2 or with UV radiation itself, these two melanoma clones both showed increased invasiveness in a transwell invasion assay. I did give parts of this talk in January, but at the same seminar, at the same.
Hey, John.
Sorry. Yep.
Hey, John, can you disable your camera to see if the sound might improve?
Am I not coming through?
You are, but you are kind of breaking down at the moment. So just shut your camera and let's try it for a few minutes, see if it's gonna work.
I don't know if I can do that.
Yep. Okay. Go ahead.
I hate to think that people weren't hearing me. You can see we're back where I was.
We could hear you. We could hear you, Jonathan. It's just occasionally the odd sentence that was broken up. Apologies for that. It's not something we've encountered before.
Sure.
Carry on.
Sorry about that. And just to be clear, you can see the slide that I'm showing?
Yes.
Okay, good. So I gave a version of this talk in January at a conference. And at the same conference, Mo actually presented some data using a new drug, IA65, that he had they had just published with Gregory Monteith as a stroke calcium entry potentiator. And so we were very interested in that. There really isn't one. You know, there's lots of things can inhibit stroke calcium entry, but I wasn't aware. I think this is the first pharmacological agent that can activate it. It doesn't truly activate it. It potentiates existing stimuli that are already activating the channel. So at the conference, Mo also put out a general call saying anyone who wanted it should ask. And it turns out that he meant it, because we wanted it, we asked, and he sent it. He sent lots of it, actually. And so we've been working with it ever since. This is essentially what we found. So it looks like IA65 does actually block or reverse the SOC suppression that we see in response to UV. And it can also reverse the SOC suppression from ORI1 overexpression, as you can see here. This is actually a WM983 line. The O1C52 is a WM983 line. It's not quite as good as the B16 ones, actually, but nevertheless, that's still a work in progress. So okay, so this IA65 reverses this. Does in fact reverse the SOC suppression we see in response to UV or ORI overexpression. Further, IA65 completely blocked the increase in invasiveness that is caused by UV. There's as you can see, the effect of ORI1 in this WM983 line wasn't quite as clear. So I'll just say that the amount of SOC inhibition in this line was quite a bit more than in the other groups that we looked at. We're really looking for another clone now to try and address this. But even in the absence of a large effect on invasion, we still see less invasiveness in response when cells were treated with IA65. Finally, we put these ORI1 overexpressing B16 clones, O1C3 and O1C44, and we did the same metastasis experiment that we did with UV-irradiated cells. Okay, and so the cells are injected subcutaneously in mice. We wait 2 weeks and we look to see if tumors form, and if they form not only in the primary tumor, but if they metastasize. And it looks like that's exactly the case. So again, none of the parental cells, all the parental cells form primary tumors, but none of the mice receiving parental cells subcutaneously metastasized. But these ORI1 overexpressing clones both did, and so we see lots of melanoma inside the peritoneum in both mouse models. I didn't comment on it before, but there wasn't any metastasis in the lungs. And that was a little confusing to us. So the typical path would be for cells to leave the tumor, enter the bloodstream, and they go into the lungs and then potentially metastasize further from there. But that wasn't the case here. And we never saw tumors in the lung, and we were wondering how it is that they accomplished it. So we got the idea that maybe they're just going right through the wall, right? And so these are subcutaneous tumors sitting on a peritoneal wall. We think what happens is the tumor cells just basically go through the wall, and we do have some evidence of that. So this is a control subcutaneous tumor, and you can see the tumor is essentially intact. There's no tumor cells even coming up close to the muscle, but you can see the ORI1 overexpressing cells or the UV-irradiated cells. There's no space. We even see some tumor cells inside the muscle itself. So essentially what we think happens is that these cells are actually breaking through the wall and entering the peritoneum in that way. In terms of tumor area, interestingly enough, the UV irradiated tumors were actually larger. I didn't comment on that earlier. I was waiting for that. They're larger, and whereas the ORI1 overexpressors are smaller. So the ORI1 overexpression seems to cause an increased exit from the primary tumor. We also see more tumor cells in the wall of the ORI1 overexpressors. So the ORI1 overexpression is increasing the exit from the tumor, but it's not stimulating tumor growth at all. Whereas UV irradiation seems to be doing both. UV radiation does things that ORI1 overexpression or stored calcium entry suppression does not, which is not really that surprising. Finally, I do have a couple of things after me finally, but I'm getting there. I want to talk about primary tumor cells. So we're associated with the Fox Chase Cancer Center, and for a number of years now, we've been getting primary freshly isolated surgical explants from Jeffrey Pharma, a surgical oncologist at Fox Chase. So these come at an unpredictable pace. We don't get any tumors for weeks sometimes, and then another week will come and we'll get the call, or to be more accurate, Scott, my graduate student, will get the call, and he has to go running over to Fox Chase, get a tumor, a freshly isolated tumor of completely unpredictable size, and then he sets up an experiment, and away we go. So we have about, looks like we probably have around 30 of them now, which is not bad. I was interested, somewhat surprised actually, that the majority of, when we looked at star-product calcium entry, the majority of these freshly isolated explants exhibited very low star-product calcium entry. I sort of thought that it would be the other way around, that we would be looking at the minority population, but it seems to be the majority. There were a few, as you can see here, that showed a large calcium response, but most of them didn't. Scott did start UV irradiating them at some point. If SOC was already low, UV irradiating them didn't really do anything, which is maybe not that surprising. We see a modest star-product calcium entry inhibition in the ones that had already had high star-product calcium entry. It's not significant, but there's only 3 of them, since it's a relatively rare thing to happen. Patient 8 was interesting. It's the only patient in which we got matched tumors from 2 different places. So we got 2 lymph nodes and 1 primary tumor. And you can see that one of the lymph nodes actually showed a significantly lower SOC than the primary tumor, which is pretty exciting in a way. I think it would be more exciting if we could get more patient samples, but there's just no way to control that. The samples come when patients are willing to share them, and that's really about it. I'll summarize what I've got so far. And so we find that UVR exposure leads to stable SOC suppression in melanoma. SOC suppression promotes melanoma progression, and primary surgical explants exhibit suppressed SOC. I want to talk a little bit about how UVR suppresses SOC, and also a little bit about how SOC suppression might drive melanoma progression. I only have a few slides for each. So this is something I've already showed you. We've observed a somewhat stable change in SOC suppression in these different melanoma cell lines. At a 2-week time point, the UACC257 had essentially no SOC suppression and no change in invasiveness, whereas the opposite was true in WM983. So what we did is we compared the genes expressed between these 2 cell lines, looking for UV responsive genes that could be responsible for SOC suppression and potentially melanoma invasiveness. Okay, and so this is the actual data, that sort of example of that data that actually went into that slide. Actually, this specific data was used, these are data generated by Scott immediately before lysing the cells and then sending them for RNA-seq, because of course, before doing the RNA-seq, he wanted to make sure that the phenotype did in fact match what we expected. So you can see that UV radiation has absolutely no effect on SOC in UACC257 cells, but a significant suppression in WM983. So this is what we found. And so there were 421 genes that were whose expression was significantly changed in WM983 cells, but not changed in UACC257. So Scott collected those genes and then did an IPA analysis. So when you do that, the top pathway that pops up is a super pathway of cholesterol biosynthesis. And actually, many of these other pathways that you see below are reflections of the same response. I mean, there's a large change in the processing of cholesterol and related lipids as connected to the mevalonate pathway. So this is the mevalonate pathway itself. It's a cholesterol biosynthesis pathway, but it doesn't. So this is cholesterol at the bottom. The mevalonate pathway drives cholesterol production, but it also drives the production of a number of other molecules. And circled are the various genes that were upregulated in the mevalonate pathway in response to UV radiation in WM983 cells. Okay. Now, some of you may realize that cholesterol has been published already the Roman lab to inhibit store-beta calcium entry. So we were interested in this as a potential reason for why calcium signals were suppressed in UV irradiated cells. So one thing I didn't want to show that. Anyway, one thing that we wanted to show was establish whether or not upregulation of the Medallion pathway was in fact causing an increase in cholesterol. The way we decided to do that is using this is not our idea. This is an idea that we've copied from somebody else. But basically, there's a bacterial toxin called perfringolysin O, which binds cholesterol and uses that binding to basically kill the cell. So the toxin binds the cholesterol and brings the toxic component to the cell. Now, domain 4 includes the cholesterol binding component without the toxin. Okay, and so you can take domain 4, attach it to a fluorophore, and then introduce that to cells, and it will bind to cholesterol and show you where the cholesterol is. And you can see that here. So we see an increase in PFO binding. This is PFO bound to M-cherry, actually, in this case. So we increase in PFO binding in response to UV radiation. That's the black bars here. Further, if we treat cells with compactin or TAC475, we see less fluorescence, or less fluorescence in the membrane. Okay, and so this means that basically UV does cause an increase in cholesterol levels, and we can block it with known inhibitors. So compactin is a statin. So the statin targets HMG-CoA reductase. That's far upstream, but it will of course block cholesterol. People take it, therapeutically. The TAC475 is much more specific. It targets the enzyme that converts choline into cholesterol. People have tried, interestingly enough, people have tried to use that therapeutically, and it turns out that people don't really like it. It's a much more specific inhibitor of cholesterol, and I would say probably a better choice in the lab because of its specificity. But for people, it's not so great, and it's a little toxic, and so clinical trials were stopped a long time ago. And so so next thing I want to show you, these same inhibitors, compactin and TAC475, they don't have much of an effect on store-beta calcium entry in control cells, but they do stimulate calcium responses in UV irradiated cells. Okay, and so the brown line is TAC475, the gray line is compactin, and the blue line is control. You can see that when UV suppresses calcium signals, and then if you treat cells with compactin or TAC475, those calcium signals are restored. Further, both agents block invasiveness. Okay, and so both cholesterol and SOX suppression seem to be important for the increased invasiveness that is observed in response to UV radiation. Okay, so UVR leads to cholesterol production, and cholesterol suppresses SOC and promotes melanoma progression. So why does this happen? This has been a burning question for us. It's been a tough one to answer, but we do have some ideas about it. I think I'm going to give you basically where we're thinking right now, and that'll bring us to the end. So we looked at metabolism. So our thinking was that this probably has something to store-beta calcium entry promotes invasiveness through the formation of invatopodia, but calcium signals are well known to modify metabolic activity. Metabolism is certainly important for melanoma, for metastasis in general, in melanoma and other systems. We were imagining when we did this experiment that you're seeing here, looking at oxidative consumption rate and extracellular acidification, that we would see something resembling the Warburg effect, where you would see inhibition of OX-phos and increased extracellular acidification, but that's actually not what we see. So both UV so either UV radiation or ORI1 overexpression both suppressed oxidative consumption, OCR, and decreased modestly in the case of UV extracellular acidification. So really quite distinct from the Warburg effect. At the time, we had our doubts. We weren't sure if we wondered if this was just done wrong or something, although this was actually done using a collaborator that was an expert in the area. So we actually measured glucose uptake and lactate production to see if it was consistent with what we were seeing. And well, yes and no. The lactate production was consistent with the fact that if there's no increase in extracellular acidification, because of course that would result from lactate production. Interestingly, we see more glucose uptake. And so what we're seeing then is that these cells are taking up glucose, but they're not they're not using it to make ATP, essentially. So they're not using it to make ATP. They're likely using it to drive biosynthetic pathways. And we were wondering a little bit about what those were. That's definitely still a work in progress. We did RNA-seq again, and this time what we were looking for was genes whose expression was up or down regulated in cells that were treated with UV and incubated for 5 weeks, but not 2 weeks, and the ORI1 overexpressing cells. And so the reason we did 5 weeks but not 2 weeks is that after 2 weeks after UV radiation in the B16 model, they weren't actually invasive yet. So we're focusing on this specific subpopulation here. I'm not going to take you through all of that. It's definitely a work in progress, but I will say that this hexosamine pathway, several members of the hexosamine pathway were actually upregulated. So this is a biosynthetic pathway that drives oglycination. So this is going to increase essentially a specific type of glycosylation on proteins in these cells. And we can show that if you block this oglycination through OSM1, an OGT inhibitor, you can in fact block invasiveness. So oglycination is required for tumor invasiveness here. I'll just summarize. So what I've shown you today, UVR suppresses SOC through upregulation of the Medallion pathway and increased cholesterol biosynthesis. We see partial SOC suppression increases melanoma invasiveness, which is distinct from, not from, total SOC blockade. And finally, SOC suppression may redirect glucose metabolism towards biosynthetic pathways to support invasive behavior. And so all this, here is Scott. You can see he's happy, and he's happy because I took that picture just when he started the lab, not recently at all. These are two technicians working in my lab. Of course, Rob Hooper initiated a lot of this work, and a long-standing collaboration with Rob Zizzi and Jeffrey Pharma that has also supported this study. Thanks for listening. Sorry if I went a little bit long, but yeah, well, that's how we're at. If you have any questions, I'll be happy to answer them.
Thanks. Thank you very much, Jonathan. I think it is safe to put your camera back in because it made zero difference into the quality of the sound, unless somebody else could hear better than I did. Anyway, there's a question that just came in from Anna, which I will start with first. And actually, I had a variation on the same questions, but let me tell you what she said. She said, if you had access to patient data, was there any correlation between patient outcome, i.e. metastasis, no metastasis, and the levels of SOC measured in these patient-derived cells? So for me, I was thinking, because you were getting those tissues, I mean, do you have at least this the surgeon removing those tumors, recording what's?
I'll get to that. I completely understand where you're going. So let me just cut it with a sort of an answer. And so we have expression data on mutations and key genes, and they're kind of all over the map. I mean, you see all of the typical melanoma driver genes mutated that you might expect to find, but no particular link to what we're showing. We also have a promise that they will give us some outcomes data on these patients, but we haven't yet got it. One caveat to that, of course, is that that kind of data. So, for example, we've collected melanomas this year, but outcome data on those melanomas wouldn't really be very useful. We do have some of those samples that were tested several years ago, and I would think that those would be really useful information, and I do want that information, and we're going to start asking for it. But it requires an ask basically, and some level of interaction with IRB that hasn't yet happened, but that is definitely going to happen.
Okay, there's a question from Rajesh Bhardwaj, and he just says, did you also see comparable UV-induced SOC suppression in normal, i.e. non-melanoma cells?
No, we didn't. The short answer is no. It's actually quite different. In fact, we don't see anything similar to that in primary melanocytes at all. It's very different. And cells were
another. Go ahead.
I have actually kind of vaguely remember that when Rob looked at hex cells, he just didn't see that inhibition. Primary melanocytes were completely different. This was I mean, we didn't look at every cell, of course, but I would say that it is somewhat of a melanoma-specific phenomenon.
Okay. So another question from Rajesh. This refers to the Btp2 data. He makes the point that could this biphasic effect of in the invasion experiments be due to off-target effects of Btp2? And he asks, did you try other SOC blockers?
That's a fair question. No, we didn't try other SOC blockers, and we certainly could. We in support of the on-target effect, we looked at ORI overexpression and saw a related effect. But we could certainly try another blocker as well. Okay. Easy enough.
And Anna Rossi again, she has a question. She said, "Great talk. Can you speculate on why SOC ablation eliminates invasiveness, whereas reduction of SOC increases it?" That's a really fair point.
Right, but so calcium. This is I am not going too far out by saying that calcium can do a lot of different things. I think there's the evidence that calcium drives invadopodia formation is extremely strong, and invadopodia are very important, are a key component of invasiveness, right? I mean, so cells will make these well, I think we know. Assuming we know what invadopodia are, I mean these parts of the cell that will reach out and then release proteases to drive invasiveness. It's an integral part of the invasive process. So I would say that these partial suppression of SOC isn't sufficient to block invadopodia formation, but total ablation of SOC does block invadopodia formation. That last part is definitely true. I mean, people have there's a lot of many other people have published this already, that if you block calcium signals, you don't get invadopodia formation. So if you can suppress calcium signals without blocking invadopodia formation, you can get a different result. Our data is suggesting that there's a sweet spot. Well, sweet is the wrong word because it's bad, but a sweet spot for SOC suppression, at least in melanoma, that allows the calcium-dependent invasive processes to go on, but also supports or drives other pro-invasive processes. In this case, we're thinking of biosynthetic pathways at the moment.
Okay. Sandip Patel asks, can you use cholesterol binding deficient ORI expressing cells, that's a mouthful, to see if they are insensitive to UV? Nice suggestion.
That's a nice suggestion. Yeah, that would be cool. I mean, we haven't done it, but that would be
okay.
That's
enough. Don Gill asks, from the summary data, I wasn't sure if the compound IA65 was increasing SOC, but perhaps I missed it.
It did. It did increase SOC. Modestly, but it did. Well, in this case, the inhibition was modest, the gray line larger than the black line, and this is the statistics on the data. So basically, if you have a 25% decrease in structural calcium entry, you can reverse that effect with IA65. In control cells, you don't see anything, right? I suppose that's because these are capsid treated cells. I mean, it should be a full SOC activation. I guess the question would be, why is the SOC being suppressed? And with the mechanism, perhaps cholesterol-dependent mechanisms that drive SOC suppression, that's what can be reversed by IA65. That's what I would suggest.
Yeah, thank you. Don asks again, the ORI1 overexpression data was interesting, but did you compare the effect of overexpressing an ORI1 mutant that cannot interact with STIM?
Well, we used the E106A, which blocks SOC, and if you do that, then the cells are completely non-invasive in vitro and in vivo. We didn't actually. We could have used a different ORI that doesn't inhibit, that doesn't bind STIM. Is that a dominant negative, or just does it just fail to be activated?
Don?
So yeah, I just thought it was can you hear me?
Again,
yeah. Yeah, I just thought it would be interesting. I mean, the overexpression, has a dominant negative effect. If it doesn't interact with STIM, it would be interesting to see whether it was through the ORI channel. But I guess the E106A, I guess I'm not sure if that's the same. It's just whether the dominant negative should not happen if it is working through STIM.
Yeah, I was asking if it's a dominant negative or is it just inert?
Right.
Is it a dominant negative? The STIM binding mutant?
If it doesn't bind STIM, then it cannot. I don't think it can work as a dominant negative, right? It's because it mops up the STIM that it works like that. Yeah.
Okay.
It's an interesting experiment, at least, because
haven't tested it, but it would differentiate between effects of ORI overexpression versus effects of SOC suppression. Yeah, that's interesting. Thanks.
Yeah, good. Thanks.
Yeah, thanks, Don. There is so in this seahorse data that you have, this is something from me. Do you have like an overexpression experiment? Is that an overexpression of E106 Yeah, here.
No, we didn't do the E106 here.
So what's those red and green traces are?
Those are two clones, two ORI1 overexpressing clones expressing different amounts of ORI1 essentially. So they both exhibit.
So do you have the
suppression? Yeah.
Say that again.
They both exhibit modest SOC suppression due to basically due to STIM seclusion. I think that would be the right. It's sequestering STIM essentially by overexpressing ORI1, sort of.
Yeah. So what do you think? I mean, I know you kind of touched on that, but this disconnect between glucose uptake and lactate production.
Well, we think that the glucose is being used for biosynthetic pathways essentially, right? And so the cell takes up glucose, and then it can really do three things. It can do aerobic glycosylation, normal aerobic glycolysis. Sorry, or it can do full-blown regular ox-phos. That can be completely eliminated to carbon dioxide, or it can be used to drive biosynthetic pathways. And then it sort of links to. I'm not sure. I don't actually think that this is the whole story. I think it's probably part of the story. But the idea would be that here some of the glucose is being used to glycosylate things and to glycolate things. But probably some of the glucose is also being used just to make other small molecules that are used to synthesize new parts for new cells or new invasive activities, essentially.
Okay, thank you. Is there any other questions?
I've got a question, Mo. So I was typing it, but given the lateness of the time and my typing's so slow, Jonathan, the SOC sweet spot that you talk about that increases invasiveness, is that also a sweet spot for cell survival?
We don't. Well, it could be, but we don't see a big survival difference. So, but I can see I certainly see the point that calcium, it could be protective for calcium overload.
Right. So if you challenge them with a pro-apoptotic stimulus, for example, then it's neither.
Yeah.
It's not a gain of function in that sense.
Well, I mean, so in vitro we don't see a big difference in survivability. All these manipulations are done without trying to kill them, essentially. So we just don't see a big difference in survivability in vitro.
It's just if there's what I was thinking was, and maybe this is kind of the wrong way around, but I was thinking that the increased invasiveness you see could be the same invasiveness but with more survival, because if the cells survive, then they'll appear to invade more. But if you don't see any difference, so that's just the thing you need to know that it is genuine invasion as opposed to a lack of death.
I didn't I didn't show it because it wasn't. It's sort of new and a little ugly, but they also migrate better in the scratch test.
Right.
I don't think it's just a matter of
sure
survival. I mean, all the stuff they do in vitro shouldn't be survival. In vivo, yeah, absolutely. You could just explain that way, but I don't see why they'd be dying in trans-well assays or in a scratch assay. If you see what I'm saying?
Yeah. No, I get you.
Any other questions? Okay. You can, well, if there are no more questions, I will just turn the baton to Martin Bootman, and he will take it from there. Thank you very much, Jonathan, on behalf of everybody, for a great talk.
Thanks.
Yeah, thanks, Mo. That's really kind. And Jonathan, super talk, really interesting. Thanks very much for coming, everybody. We'll do this all again in 2 weeks' time. We're going to take a break for Christmas, but we'll be back in the new year. We'll advertise that program on the spreadsheet sometime soon. But for now, thanks to everybody for the great questions, for the brilliant talk, Jonathan. And stay safe.
Martin, there's one more seminar coming. There are more seminars coming, so I might not have listened properly.
Yeah, sorry, we haven't broken for Christmas yet. We will take a break at Christmas, but thanks, Katja. Yeah, we've got Kevin talking next time. So please do come along in a fortnight, and hope to see you then. Stay safe, everybody.
Thank you.
Bye now.
Bye.
Bye. Cheers.
Bye, everybody.